Hello guys, welcome to this video of biology. So for today I'll be looking at plant nutrition, everything to do with plants and how they feed. So we start with photosynthesis, a very common subject. This is a process by which green plants manufacture glucose or starch or carbohydrates from carbon dioxide and water in the presence of light energy absorbed by chlorophyll. Okay, that's photosynthesis. Oxygen is produced as a byproduct. This process takes place in leaves and may be summarized by the following word and chemical equations. So it takes place in leaves, and this word equation is what summarizes the whole process of photosynthesis. We can see carbon dioxide reacts with water in the presence of light energy on chlorophyll to form glucose and oxygen as byproducts. And this is the chemical equation: 6CO2, that chlorophyll or light energy, we have glucose plus oxygen. This is a balanced chemical equation for photosynthesis. So the products for photosynthesis are glucose and oxygen. The oxygen is released out of the plant, or some of it is used for respiration. The oxygen is released out of the plant while some of it is used for respiration. That's why we say plants are important to us, because they release oxygen, and we know that we need oxygen for us to breathe and survive. So the plants release oxygen out of the plant, and some of it is used for their respiration. The glucose formed is metabolically active and takes part in the following reactions. This is the glucose which is formed. So the glucose, some of it is used for respiration, some of it is converted to cellulose and becomes part of cell walls. That's the glucose. Some of it is combined with nitrogen and used to synthesize amino acids. Some of it is combined with nitrogen and used to form or make amino acids. And some of it is converted to sucrose in order to be transported. Okay, some of it is converted to fats and oils. Some of it is converted to nucleic acids. Some of it is converted to nucleic acids. The excess is converted to starch for storage. So in plants, glucose is stored as starch. It's converted to starch. While in animals, it's converted to glycogen. It's converted to glycogen in animals. But in plants, it's converted to starch for storage. So glucose takes part in these reactions. Okay. So light and dark reactions. We go to light and dark reactions under photosynthesis. Light reaction. During this stage, light energy by chlorophyll is used to split water molecules into oxygen and hydrogen. This is called photolysis. Oxygen diffuses into the atmosphere while hydrogen proceeds into the dark stage. Okay, I repeat, during this stage, light energy absorbed by the chlorophyll. So the light absorbed by the chlorophyll is used to split water molecules into oxygen into oxygen and hydrogen gases. This is called photolysis. Oxygen diffuses into the atmosphere while hydrogen proceeds into the dark stage. This is a light reaction, dark reaction. During this stage, hydrogen from the light reaction combines with carbon dioxide forming glucose. This is at night now. During this stage, hydrogen from the light reaction, the one which was left in the light reaction, combines with carbon dioxide forming glucose. So carbon dioxide combines with hydrogen to form glucose. Okay. Note, one thing we should note is that carbon dioxide comes from the atmosphere by diffusion. Carbon dioxide comes from the atmosphere by diffusion, and water from the soil by osmosis. Water from the soil by osmosis. Okay. To determine whether photosynthesis has taken place, the leaves of plants are tested for starch. The steps involved in testing a leaf for starch are: number one, you boil the leaf in water for the purpose of killing the protoplasm and making it permeable to iodine solution. Okay, so you boil the leaf in water. Number two, you boil the leaf in alcohol using a water bath. This is to extract chlorophyll so that it does not interfere with color changes. A water bath is used because alcohol is highly flammable. However, the alcohol also makes the leaf brittle. Okay, that's point two. Place the leaf in warm water to soften it. This is the process. Spread the leaf on a wide towel and add a few drops of iodine solution. This is to test for starch. The iodine solution turns blue-black. Starch is present, and if it remains yellowish brown, then starch is absent. So that's how we test for starch. If the iodine solution turns blue-black, then starch is present. If it remains yellowish brown, then starch is absent. That's in plants. Okay, we go further to the requirements of photosynthesis. What we need for photosynthesis to take place. These factors that need to be present for photosynthesis to take place are carbon dioxide, water, sunlight, and chlorophyll. Those that also affect the rate of photosynthesis are limiting factors of photosynthesis. Carbon dioxide enters the plant by diffusing through small openings. So carbon dioxide enters the plant by diffusing through small openings in the leaf called stoma. That's the singular, or stomata for plural. The small openings. Water enters the plant through the roots by osmosis and moves up the plant through xylem vessels. Water enters the plant through the roots by osmosis and moves up the plant through xylem vessels. Okay, sure you understand that. Light energy, which is mainly solar energy from the sun, is captured and trapped and stored by green pigment called chlorophyll. Found in the chloroplasts. So the chlorophyll is found in the chloroplasts. During photosynthesis, this solar energy, which is caught, which is caught by the chlorophyll and stored in the chloroplast, is transformed into chemical energy Since photosynthesis is an enzyme catalyzed reaction, so it forms into a chemical. It's transformed into a chemical. Since photosynthesis is an enzyme catalyzed reaction, its rate gets affected by all factors that affect enzyme activities. So the rate of photosynthesis is affected by the same factors that affect the rate of enzyme activities. Okay, that's one thing you should note. Now we look at an experiment to show that carbon dioxide is necessary for photosynthesis. Let us show and prove why carbon dioxide is necessary for photosynthesis. Discharge a well-watered potted plant by placing it in the dark for at least 24 hours. During this time, all the starch present in the potted plant is used up. Okay, set up the experiment as shown in the following diagram. So you can see the one leaf is placed in a conical flask of containing sodium hydrogen carbonate or distilled water. The other leaf is placed in sodium hydroxide. Okay, let's go further. Place the potted plant in sunlight for 4 to 6 hours. Test leaves A and B for starch. Leaf A turns blue-black, showing the presence of starch, while leaf B turns yellowish-brown, showing the absence of starch. This shows that carbon dioxide is necessary for photosynthesis. Let's look at an experiment to show that light is necessary for photosynthesis. Number one, discharge a well-watered potted plant by placing it in the dark for 24 hours. For at least 24 hours. During this time, all the starch present in the potted plant is used up. Set up the experiment as shown in the diagram. So now we're testing for light, proving that light is necessary. So an aluminium foil here was covering the leaf of the plant with paper clips, and we have the exposed parts of the leaf. So that's the setup. Place the potted plant in sunlight for 4 to 6 hours. While the plant is in sunlight, draw the selected leaf showing the exposed parts and covered parts. Test parts A, or the exposed part, and B, the covered part, for starch. You note that part A turns blue-black, so the exposed part turns blue-black, which shows the presence of starch. While part B, which is the covered part with aluminium foil, turns yellowish-brown, showing the absence of starch. This shows that light is necessary for photosynthesis. So another experiment to show that chlorophyll is necessary for photosynthesis. Discharge a well-watered potted plant which has variated leaves by placing it in the dark for at least 24 hours. During this time, all the starch present in the potted plant is used up. Just like in the other experiments, place the potted plant in sunlight for 4 to 6 hours. While the plant is in sunlight, draw a selected leaf showing the green parts so that they can easily be identified even after chlorophyll has been removed from the leaf. Label the green plants as A and B. As A, label the green parts as A. and the white parts as B. Okay, test the parts A, which is the green part, and B, which is the white part for starch. You will see that part A turns blue-black, showing the presence of starch, while part B turns yellowish-brown, showing the absence of starch. This shows that chlorophyll is necessary for photosynthesis. Okay, so these are experiments to show why chlorophyll, carbon dioxide, and light are necessary for this process called photosynthesis. So we go to measuring the rate of photosynthesis. This can be measured by counting the number of oxygen bubbles produced by an aquatic plant. An example is pondweed. And Elodius sp per unit time. A typical setup for such an experiment is shown in the following diagram. So you can see the pondweed, the water trough, and the oxygen bubbles, as well as the inverted funnel, the water, and the test tube. So if we count the number of bubbles, we can see the rate of photosynthesis just by counting these bubbles. So the rate here is 1, 2, 3, 4. 4 sp per unit time. Okay. So next we go to the leaf structure. So the leaf has got 1, 2, 3, 4, 5, 6 main structures, which are the leaf apex, the tip. It has got veins. It has got a midrib, which looks like a stem of a leaf, called the midrib. We have the leaf lamina. We have the leaf margin. We have the leaf stalk or petiole. Okay. Now we look at the cross-sectional structure of the leaf. So in the cross-sectional structure, we have the cuticle, we have the upper epidermis, we have the chloroplasts, we have the palisade mesophyll. Okay, so the cuticle is a very smooth part of the leaf. That smooth part of the leaf. The upper epidermis seemingly like a skin. It's like a skin of the leaf. The chloroplast, you know what the use of the chloroplast is. You have the palisade mesophyll. Okay. So just to finish up photosynthesis, I'll look at adaptations of leaf for photosynthesis, as well as the importance of photosynthesis and applications of photosynthesis in greenhouses. And I'll call it a day or a night, whatever time you're watching. So very quickly, I go to the adaptations of photosynthesis, of the leaf, in photosynthesis. So a thin lamina is required for easy penetration of light. Large surface area to capture as much light as possible. Presence of veins or vascular bundles to supply the leaf with water, the xylem, and to transport end products of photosynthesis, the phloem. Presence of stomata for entry of carbon dioxide and exit of oxygen by diffusion. Presence of chloroplasts to absorb light energy for photosynthesis. The highest concentration of chloroplast is found in the palisade cells, followed by the spongy cells, and finally the guard cells. These are the adaptations of a leaf for photosynthesis to take place. The importance of photosynthesis is that it produces food for all organisms directly or indirectly. It maintains a balance, equilibrium of oxygen and carbon dioxide in the atmosphere by using carbon dioxide from animals and producing oxygen for animals. It produces a vast amount of energy in woods, peat, and coal, etc. These are the importances of photosynthesis. Applications of photosynthesis in greenhouse. A greenhouse is an enclosure with walls of transparent glass or plastic where plants are grown. By having transparent walls, light and heat are allowed to reach the plants. In some green houses, plants are supplied with artificial light from electric bulbs. The walls minimize escape of heat from the greenhouse, thereby keeping temperatures high inside the greenhouse for optimum enzyme activity. Sometimes the greenhouse is artificially supplied with carbon dioxide. These factors make a greenhouse more productive than an open piece of land. Okay, so this is how photosynthesis is applied in a greenhouse. Thank you so much for watching this video. We've looked at photosynthesis. Under plant nutrition. So next time I'll continue with the same photosynthesis, but I'll be looking at the plant storage organs. Thank you so much for watching. Make sure to like and subscribe.